Welcome to Lesson 32, Autoclave Validation. In this lesson, we will be looking at aseptic processing, steam sterilizing, autoclave operation, control of sterile production, and the validation of the autoclave, including a Bowie-Dick test. Aseptic processing overview. So we talked earlier about the two categories of sterile products, those that can be sterilized in the final container, terminally sterilized, and those that cannot be and need to be aseptically prepared. In this lesson, we're going to be talking about steam sterilization. So for moist heat, we talked again earlier about the two configurations. We had steam out of place, SOP, for portable items, and this is where we would use the autoclave. The second type was the steam in place, the SIP, for fixed equipment, and that needs to be connected to utilities in order for that to function. So steam in place. This is where we repeatedly steam areas of product contact, including vessels, flow paths, and sample ports. This may be done to reduce the bioburden on the system or to kill harmful materials at the end of a batch. A typical steam-in-place system will ensure that all areas being steamed have been exposed to live steam for an adequate time to make sure the desired kill effect takes place. The steam system must be able to measure and to control steam temperature, pressure, and/or the flow to ensure that there is adequate steaming. The calculation of the F0 or other measures is often required to meet the process needs. So when we look at this diagram, you can see the area in blue is our water, and the area in orange is where we've got superheated steam, and the line between the two is where our steam saturation line is. So if we look at 1 bar of pressure, which is equal to 15 psi, that's giving us a temperature where where our steam saturation is at 121 degrees, and that's where that number comes from. This is the working of a steam sterilization autoclave. So this is what it generally looks like. So the first thing we would do is we'd load our items inside in it and seal off the door. After that then, the steam supply would be initiated and you'd have the steam coming in. At the same time, you create a vacuum to pull out the cold air. There's a safety valve always associated with it that if the pressure gets too high, the door will open. And we monitor the pressure throughout to make sure that it is operating efficiently. Autoclaves look something like this. You can have them on all different size scales, from a small lab scale autoclave to a large industrial scale one, and then even larger a biopharma scale where you might have two sides to it. So you'll have a non-sterile environment, and then you might have your products exiting on the other side to a sterile environment. The operation of an autoclave. So steam is less dense than air, and it will rise to the top of the autoclave. This will eventually push out the air through the port at the bottom. A vacuum is also used then to pull the air off and to pull off any condensate that may be there as well. Air is very poor at reaching sterility. Steam at 134 degrees Celsius can achieve in 3 minutes the same sterility that a hot air at 160 degrees takes 2 hours to achieve. Run through the cycle. So the vacuum removes the air, even the steam circulation out to allow contact with all the surfaces. The steam feed is a clean steam, obviously. It's ramped up to the temperature and the pressure required. And that temperature is maintained and that pressure is maintained for a specific time. Generally, we're talking about 121 degrees C for 15 minutes. Then there is a cool down or a pressure ramp down, where you have an addition of compressed air or cool water to speed it up. And then your cycle is unloaded. You would then confirm the effectiveness of your cycle and proceed. Control of sterile production. Separation between sterilized and not yet sterilized products is important. We have to remove the risk of any mix-ups and cross-contamination from potentially occurring, as these would be disastrous in a sterile environment. Each basket, tray or other carrier has to be properly labelled, with the name of the material, the batch number, the sterilisation status. Sterilisation records for each run are approved as part of the batch release procedure. They are so important. The loading plan must be developed and rigidly followed to ensure adequate space for steam circulation. Sterilisation tags and tapes look something like this. So you'll have batch sterilisation tags and sterilisation tapes, and you can see that they will change colour based on whether they pass or fail. How we validate an autoclave. The things we need to verify. The sterilisation conditions have to be correct in the sterilisation cycle. That the sterilisation cycle will consistently fulfil its purpose. They are the main goals of a validation for an autoclave. How do we do that? Well, we look first of all at the system impact assessment. So our design review, we need to know what we need it to be capable of, what size we need it to be, and what dimensions design we need it to be. When we install it, we'll make sure that it fits, that all the components are present, etc. Operational qualification. We're going to check all parts of the autoclave operate correctly. Do the alarms work properly and the interlocks? Can we do a pressure and a vacuum test? Are they both effective and working correctly? Safety checks, are they all okay? Drainage and electrical power testing, is everything hooked up all right? Then we would look at the extremes of the operating conditions, the highs and the lows. Does it do what it's supposed to do? We would do some heat distribution studies then of an empty autoclave, so we would be looking for the cold spots in the autoclave. For our performance qualification, we would look at the heat distribution of a filled autoclave, so how does it work when it's full? We would also look at heat penetration studies. We would verify the loading using biological and chemical indicators that we saw on our previous slide. If applicable, we would generate the max or the min load capacity. So for example, if you had 15 bottles in your maximum load and 2 in your minimum load and you validated both, it gives you capability to run other volumes in between once you have validated it and the load pattern that goes with it. We will generate our F0. We'll generate our SOP and we'll make sure that all our personnel are trained and ready to go. We would then run 3 consecutive runs to show that it is operating consistently. This is an example of what a loading pattern looks like. So a loading pattern is how you fill your autoclave. The items must be placed in the autoclave according to the validated loading pattern. Any changes to that loading pattern would require revalidation. This is what a typical liquid load qualification might look like. And you can see all those wires coming out of them. They're all the thermocouples being used for a heat study. All the load configurations then need to be studied. Thermocouples, they measure your temperature, and the biological indicators, the colour changing tapes and packs that you saw in the previous slide, will be used for this study. We will always put one close to the drain because that is the worst case area for coldness. That is your cold spot in your autoclave, because that is where your cold air is coming out. The biological indicators removed from the load are following each study. They're placed in a sterile container and submitted to QC microbiology for analysis, as per the relevant microbiology procedure. This is what the plan for your thermocouples inside in your empty chamber might look like. So you see TC 1, 2, 3, 4, etc. That stands for thermocouple 1. So that's the location of where you put them at. And you'll see that you have one at the center, in the geometric center, and you have one at the drain. They're the 2 key ones to get for your worst case areas. Biological indicators. Biological indicators have live spores, known to be the most resistant living organisms to the lethal effects of the particular sterilization process. Steam sterilization uses Bacillus stauothermophilus spores. Heat sterilization uses Bacillus subtilis spores. Biological indicators available in different formats. You can get them in vials, in papers, etc. So you would place the biological indicator in a product or in the product packaging. And this inoculation package is placed in a sterilizer and processed. Then we're going to test it in the microbiology laboratory to prove that all the spores have been killed and hence your sterilization process is working. The Bowie-Dick test theory. A technique in sterilization that ensures that the residual air in the autoclave has been removed before use of the equipment. And obviously that is important, otherwise you will have cold patches during your sterilization cycle. When air or non-condensable gases are trapped within a load, it generally forms a bubble or a pocket. The temperature in this region then is generally lower than that of the surrounding steam environment, which is a problem for us. The temperature depression caused by the air will gradually disappear as the air heats up and is downwardly displaced until it's no longer detectable. This normally takes 2 to 4 minutes. If a temperature of 121 degrees C is not achieved for a period of 8 minutes, then the colour at the centre of the test pack will not be changed to a dark. We'll have a look at what that means and what that looks like in a second. Steam with a dryness factor of 1 or greater may contribute to superheating within the chamber. If the superheat is excessive, the test pack may have a smudge or a blurred appearance around the edges. So this is what it looks like. So you can see it's yellow before use. If it's dark blue in the centre, it means it's passed. If it's still yellow in the centre, it means it's failed. And again, darker navy, then pass over 48 hours. The Bowie-Dick test requirements. The test pack includes a series of sheets of paper with a chemical indicator in the centre like we've just seen. The autoclave is already at operating temperature when the timing starts. The test pack is suspended 100 millimeters over the chamber drain. Dedicated Bowie-Dick cycles with the exposure time of 8 to 8.3 minutes and a longer time than that invalidates the test. When the cycle is complete, the test pack is immediately removed from the chamber and it's stored in a cool, dry, dark place away from sunlight once the test is completed. Z values. The Z value is the number of degrees of temperature change that are necessary to change the D value by a factor of 10. An example of a D value. So if you have a D of 121 and it takes 1.5 minutes for 1 log reduction. So your Z value is 8 degrees C. 121 minus 8 gives you D of 113. So temperature of 113 will take 15 minutes, i.e. so that's 1.5 from above multiplied by 10. So that's your factor of 10. The F value is the number of minutes of the effective sterilization cycle. It will differ from the actual length of sterilization cycle due to varying conditions of temperature and saturated steam exposure. The actual sterilization cycle time will be different as it takes the length of time above and below the temperature set point into account. The F0 value is the effective exposure time at 121 degrees C that has a Z value of 10 degrees C. For large volume parenterals, guides state that the steam sterilization process should be sufficient to produce an F0 value of at least 8 minutes. Sterility assurance level, your SAL. Sterilization is the act or the process that destroys or eliminates all forms of life, especially microorganisms. Microorganisms of the same species vary in their resistance to chemical and physical agents, and even if 1 in a billion organisms survive, there is a possibility of rapid multiplication and contamination. Remember, we are looking for a SAL of 10 to the minus 6, 1 in 1 million, to demonstrate sterility. And you remember from our previous lesson, our sterilization is 10 to the minus 6. F0. F0 value can be calculated based on the temperature recorded by the thermocouple inside a product container at the coolest area of the load. The continued verification of an autoclave. So it's an ongoing process. It doesn't stop the day the protocols are signed off for your validation. The daily tests that are done when an autoclave is being used is the preheat test. The vacuum will be tested daily. The Bowie-Dick test will be done daily, and the cycle acceptance criteria will be done, whatever that has been set for the particular autoclave. On a weekly or monthly basis, then the air detector will be tested. There'll be filter integrity testing, filter sterilization, and the preventative maintenance program, whatever has been set out in the site SOP. Just to recap the kinetics of death for steam sterilization. Your D value is your time in minutes required to reduce the microorganism population by 90% or 1 log unit. So an example of a D value, D is 121. It takes 1.5 minutes for 1 log reduction. F value, the number of minutes of the effective sterilization cycle. Example of an F value, 15 minutes. Z value, the number of degrees of temperature change necessary to change the D value by a factor of 10. Example of a Z value, 8 degrees C. The F0 value, the effective exposure time at 121 degrees C with a Z value of 10 degrees C. Here's some references and some further reading for you on autoclaves and steam sterilization. This will be listed in your appendix to this lesson plan. Over to you. What do SOP and SIP mean in relation to moist sterilization? How does an autoclave work? What considerations would you take when performing a PQ on an autoclave? Thank you very much for your time and attention. We'll see you for the next lesson.